If cancer can adapt faster than the drugs that you're taking work, you need to know why, and you need to know in advance if your cancer is like that. And the answer isn't about mutations. It's something called extrachromosomal DNA, and most doctors were never taught about it. Knowing whether this applies to you tells you how aggressive your cancer will be and how aggressive you need to be against your tumor. This episode explains how cancers adapt so incredibly fast, how they can control their own growth, and also why the experiments from the 50s that justify the metabolic hypothesis of cancer don't mean what you think they mean. So stick with us. This one's a very important episode about unique cancer biology and how it impacts the way that you have to plan your cancer treatment.
Hello, and welcome to Elevating Cancer Treatment, where we explain the science and debunk myths to help you navigate your health journey. My background is a little different. Beyond educating about cancer, I'm actually designing new drugs that are defining the future of oncology. This direct, hands-on experience offers me a very different perspective of how these cancer treatments work on the body, interact with the cancer cells, and cause side effects. And these are insights that I'm excited to share with you. If that sounds interesting, make sure to like this video, subscribe to the channel, and hit that notification bell so you never miss an update. And please share it if you find it useful. I'm Dr. Jay Chaplin. An important reminder, I'm a PhD, not an MD. The information in this video is education, and it's not medical advice. Every cancer is unique, and no general information applies to everyone. Please remember that. Always consult with your healthcare provider for guidance on your specific situation. And two quick things. First, as a thank you for being here, I've created a free resource, 10 Things to Elevate Your Chemo Journey, which you can download from the link below. And second, by signing up, you'll also get updates on that innovative cancer treatment I'm working on. I'm confident it represents a significant advancement in immunotherapy. So please take a moment, download your free guide, and join us in shaping the future of cancer treatment.
So I've said it here a bunch of times, and I'm going to continue to say it because it's true. Cancer adapts incredibly quickly. And when you apply pressure to it, when you put it under stress from chemotherapy or radiation or immunotherapy, some other approach, the cancer adapts, it changes. It's like the Borg, it gets resistant to what you're doing. And it can become resistant to drugs very, very, very quickly. Much more quickly than we would ever expect for a normal cell in your body, or even bacteria, to evolve and change. That's really impressive and very problematic. How does it do that? And how do you know if your cancer is like that? So you might have heard that mutations make your cancer stronger. But in this case, that's wrong. It's usually not through mutations. So mutations are where basically letters in the words of your DNA are changed. And that can be good, it can be bad. It's usually bad. It takes a long, long time. Acquiring mutations is normally a very slow process. Even when DNA repair pathways are already damaged, like with the BRCA or POL mutations that cause more mutations to happen, it still takes a long time. And that's because most of the time, these random mutations are not good for the cancer cells. They actually cause a lot of damage rather than making the cancer stronger. If you think about Spider-Man and Peter Parker, you've got this one case where somebody got bitten by a radioactive spider and they got superpowers, and how many other people just keeled over dead? Probably 1,000. Your odds aren't good. Sometimes it works out, but usually not. So there are even targeted therapy drugs designed to make tumors have more mutations and kill themselves off in the process. And those drugs are actually really good and very effective. So even though we normally see several mutations, anywhere from 4 to hundreds, in a cancer, that's usually not the primary way that the cancer is adapting and changing quickly. Mutations can drive growth, but they're a slow process of building those up. There's a different mechanism. And to understand that, we actually need to go back a little bit. We need to go back to how DNA is actually managed and packaged inside of your cells. So let's talk about that. Let's talk about how DNA is managed and regulated inside of your cells, and some of the ways that things can go sideways, really sideways, especially in cancers. When we think about our DNA, most of us don't have any clue how much of it there is. There is an enormous amount of DNA inside of each and every single cell of your body. Just the first chromosome, chromosome 1, just one copy of it. You got 2. Take one from one cell and stretch it out. It would be 3¼ inches long. To give you a visual, that is about the length of the short side of a credit card, or in this case, a Costco card. Or again, for those of you like me who prefer the metric system, that's about 8.2 centimeters. If you took all of the DNA from one cell, every chromosome from one single cell, and stretched it out end to end, you'd have a span of 2 meters. That's about 6.5 feet. That's longer than most human beings are tall. That's a tremendous amount of DNA in just one of your 30 trillion cells. Now, here's the miraculous bit of this. Every time a cell divides, every time it tries to go from one cell to 2 cells, it has to double the amount of DNA by carefully copying every bit of it, and then it has to faithfully segregate it. It takes one set of chromosomes, pulls it to one side of the cell, the other set to the other side of the cell, and then these 2 cells pinch themselves off and go their own separate ways. So you got to separate those. And this is in this giant spaghetti mess inside the nucleus. You've got a bunch of chromosomes. They're all tangled together. So that is an incredibly difficult process. And normally it's managed really, really well. However, in some cases, that process goes wrong. That's the first step that we have to talk about here. That can cause a huge downstream consequence. And this is where, prepare yourself, it's an awful word I know, Chromothripsis comes in. That is a process where the segregation, the pulling apart of those chromosomes doesn't happen properly, and one or more of the chromosomes get shattered. They literally get pulled apart, sometimes into hundreds, sometimes thousands of different pieces. That is a catastrophic error. It's an enormous error. And if the cell is healthy and it's acting properly, and it has a major catastrophe like this, it would decide, I can't repair this, I can't do this well, it's gonna be a mess, I'm just going to die. That's the right thing to do. And it would eliminate itself. But in a cancer cell, especially if you've already gotten rid of the tumor suppressor genes that help keep these things from happening, genes like TP53 or BRCA or something else like that, then the cancer cells just ignore that massive danger signal and they will happily do a really bad job of repairing it. So think about a book. It's got a table of contents, it's got chapters in here. Everything is in its proper place. Sorry, Dan. And now you've got chunks of DNA all by themselves. This can be very problematic for you if this is your situation, because it leads to extrachromosomal DNA. So, what happens next? One kind of error is to stitch together pieces of chromosomes. Let's just swap those. Stick them here instead of here. Yeah. Where pieces get stuck together the way they shouldn't. And people used to look for that. It does happen in some cancers, particularly leukemias. They used to look for fusions, 2 genes that normally weren't close to each other that ended up attached to each other. Kind of like you're reading along and chapter 13 just ends and all of a sudden you're reading chapter 17. Just jump straight into it. Those can lose their normal regulation, and the genes that are fused together can be permanently switched on or permanently switched off, and that can lead to out-of-control growth or really weird behavior of the cell. This does happen sometimes, but it's actually fairly rare. Why? Chromosomes are like long, wordy, boring novels with occasional good chapters sprinkled in. Not like this book. I got a bunch of these. Let's say it's an 800-page book with only 50 pages of actual action or meaningful content. When you put pieces together randomly into a book like that, and you get a weird junction, it's probably going to be in the boring part, and it's not going to affect your reading much. It may not make a lot of sense, but you're not going to be totally confused. It's not going to be super jarring, because you didn't care about the boring parts anyway. That's what tends to happen more often than fusions that directly cause problems. Most of the chromosome pieces get stitched back together again, and the buffer DNA in between the genes is where most of those fusions happen, causing relatively little impact to the parts that get put back into the chromosome. So these ones end up working fairly well, usually. Still be a problem sometimes. But occasionally, some of those pages, they get lost on the floor. They get stuck under the couch, kicked around, you find them later. What happens, your cell doesn't like DNA with ends. So chromosomes already been put together. It'll just stitch the ends together, because it's too far away from the chromosome or too late to get reattached. It just makes little tiny circles of DNA that don't get managed the same way, and they don't get pulled apart evenly like chromosomes are. The mechanism just doesn't work. So when that cancer cell tries to divide again, The copy number can change. You can end up with more or less. And all of those regulatory pieces around the gene, that boring buffer that determines whether the gene in the DNA should be on or off, have probably been stripped out. These little circles of DNA have gone rogue, and now there's no table of contents, there's no location within the book, and there's no way to tell the context. It's just off by itself. It's just its own little random piece of information. But guess what? It still gets copied. This is extrachromosomal DNA. Here's your chromosome. It's been put back together, maybe badly. This is outside. It's extrachromosomal, and that can be a big problem in two different ways. Those genes can easily be deleted, or they can be massively amplified. Instead of having only 2 copies of a gene per cell, the way that normally happens with a chromosome. So, the first way, it's a deletion. If the gene on the extrachromosomal DNA is something that makes the tumor sensitive to a particular kind of chemo, or slows down growth, when this cell divides, let's say 2 copies go over here and none go over here, this cancer cell loses these 2 copies. It has a big growth advantage. Maybe it's resistant to chemo, maybe it doesn't care about the chemo anymore, maybe it grows faster. This cell will grow faster and take over. The second way, going from red to green, is if there's a gene on one of these little snippets of circular DNA that is beneficial for the cancer. Maybe it gives resistance to chemotherapy, or maybe it gives a growth advantage. Then cells that end up with more copies of this make more copies of this. Many thanks to my lovely daughter for making these audiovisual aids for us. And more copies of this. And before you know it, the cell has an enormous growth advantage, and it will take over. Sometimes a key growth-driving gene or cancer-causing gene, what we'd call an oncogene, may not be mutated at all. It may be totally normal sequence, but if you've gone from 2 copies to thousands of copies, it will still drive massive growth and very aggressive cancer, even though it has a normal gene sequence and hasn't been mutated. Now, I know that was a lot of background info, and probably boring science for some of you. For good or for bad, I love it. But I wanted you to know how incredibly damaging this can be, and in multiple ways. It's important for what comes next. So, how likely is this, and how can you tell if it's the case with your cancer? How common is this really? Is this something that happens in 1 in 1,000 or what? Why should I care? This process where chromosomes get shattered and pulled apart and then badly repaired, researchers used to think it was very, very rare. We know it essentially never happens in regular healthy cells. The percentage of that is far below 1. It's essentially 0. That's because normal healthy cells just choose to die if this happens. They know they can't fix it well enough. But in cancer cells, researchers used to think that Chromothripsis only occurred in 2 to 3% of cancers. And that's because people were looking for obvious gene fusions, things that drove the cancer to become what it is. But when researchers started looking at complete DNA sequencing, a lot of those buffer regions in between genes are very hard to sequence. Once they got that and started looking at it carefully, what we actually see are that there are many fusions in regions in between genes, and often huge copy number variations. Instead of 2 copies, like we would expect in every chromosome pair, you're going to have 1 in each one, 2 copies. You might have thousands of copies, or 0 copies, complete deletions. And when people started looking at it this way and following up carefully, they found that the rates of chromosome shattering and repair are somewhere between 25% and 50% in cells taken directly from cancer patients. Now, that's a big range, 25 to 50%. It depends on what kind of experimental technology is used. It's probably closer to 50%. But either way, that's a very high number. Even 25% is much higher than 0. A quarter to a half of all cancers have gone through one of these chromosome shattering events and tried to repair themselves. Now, stick with me, because this is going to affect how you interpret your test results and allow you to see if you have this issue. Now, these tiny circles of DNA, this extrachromosomal DNA, it's also quite common. This is a relatively new field, looking at them. Over the last 10 years or so, people have started to look at this much more carefully. And across all cancers, about 17% have been detected as carrying extrachromosomal DNA. But that's the average over thousands and thousands and thousands of different tumors. That's the average. The rate goes up quite a bit in aggressive cancers. Remember, these can drive aggressive growth and drug resistance. For instance, esophageal cancers go from 4% in high-grade dysplasia, which is relatively mild, to 43% with advanced disease. Neuroblastomas, which are fairly aggressive, have about 30% incidence of extrachromosomal DNA. Glioblastoma is nearly 100%. And this extrachromosomal DNA often isn't even in the nucleus, where DNA is supposed to always be. It forms little tiny nucleus-like compartments called micronuclei. It's no longer a bound book in a library or bookstore in some central location, but some trashy tabloid that's in every checkout aisle in every grocery or convenience store across the entire country. You can actually see them. You can actually see these micronuclei if you stain for them. They're very commonly found in cancer cells, and especially in cancer cell lines used in the lab, ones that have been grown for decades in culture. They've had plenty of time to amplify their extrachromosomal DNA and make tons of these little micronuclei. We've known about these for a long time. We've known about them essentially for as long as people have been studying cancer, but people used to ignore them. They'd say cancer is weird, it's just debris, it's part of the cell dying. They're not debris. They are active and they are driving the biology of the cancer cells. They often have more control over the cancer cell than the DNA that's still in the chromosomes and stuck in the actual nucleus. So, bring this back to you. Your test results. This extrachromosomal DNA in the micronucleus gives cancer the ability to adapt incredibly quickly, either by increasing the copy number or decreasing the copy number of crucial genes. It lets it adapt like that. So how do you tell if your cancer has extrachromosomal DNA? These are the kinds of things that I look for when I'm looking at a client's genomic panel. I look at the panel not just for mutations, because those can tell what kind of drug might be useful, but also for copy number variation, or CNV. Because that tells me whether a gene has been amplified through this extrachromosomal DNA process. It tells me a lot about how aggressive this tumor is likely to be. If you've had a genomic panel, especially if it's next generation sequencing, which is very common these days, you can look at the copy number variation. If there's a big difference in copy number variation, that gene is almost certainly on extrachromosomal DNA. What do I mean by that? What do I mean by large difference? The number is supposed to be 2. Normal chromosomal DNA, you have 2 copies of each gene. So if your copy number is between 1 and say 3 or 4, that's relatively normal. If it's outside of that range, 0 copies means the genes have been deleted entirely. Might be extrachromosomal DNA, might not. More than 10 copies, that means you got non-chromosomal replication. That tells you that your cancer can adapt very quickly and it needs to be controlled very thoroughly. You don't want to give a cancer like that time to adapt. You want to treat it as quickly and as thoroughly as possible. I also work with clients to use a combination of different treatments to hit cancers like this from very different angles, to reduce resistance, to keep resistance from forming. So again, you can always reach out to me if you want help with interpreting your genomics panel and planning the best path forward for your treatment and making sure that you don't give your cancer a chance to adapt. Okay, the metabolic theory and why it falls short. I want to switch gears. There is a hypothesis. It's not a theory. Theories have solid support and haven't been disproven. There is a hypothesis that cancer is actually caused by the mitochondria. It's driven by the mitochondria rather than stuff that happens in the nucleus. That all those mutations in the nucleus are irrelevant, that it's the mitochondria. This comes primarily from experiments that were done in the 1950s through the 1970s by the Briggs lab. We've got links down below. They did nuclear and cytoplasmic transfer experiments. They would take a cancer cell and a normal cell, and they would move the nucleus from the cancer cell into the normal cell. Sometimes it caused cancer, sometimes it didn't. They would take the nucleus from a normal cell and put it into a cancer cell. Sometimes it would revert to normal, but a lot of times it would go cancerous. So the assumption must be that something other than the DNA, mitochondria, was doing it, because they didn't believe there was any DNA outside of the nucleus. If the nucleus didn't transfer it, if the body of the cell was the reason why there was cancer, then they thought it must be the mitochondria because DNA is only in the nucleus. The problem is they didn't know about extrachromosomal DNA at that time. They were ignoring it. They didn't know about micronuclei at that time. They thought it was just debris. They ignored it. We now know those micronuclei floating through the cytoplasm all over the place contain many, if not most, of the cancer-driving genes. So yes, if you transfer a normal nucleus into a cancer cell, it'll stay cancerous. Yeah. because all of the extrachromosomal DNA still got left there. The nucleus, the chromosomes, aren't doing most of the damage. It's the extrachromosomal DNA. And now that we know this, the interpretation of those experiments completely changes. Those micronuclei contain hyperactivated cancer-driving genes, sometimes thousands of copies. That's the driver. It's not the mitochondria. We'll talk about mitochondrial mutations and errors in mitochondrial metabolism in a future episode, but that's cancer biology that I'm interested in. That's not necessarily relevant to you. I just wanted to let you know how micronuclei and extrachromosomal DNA do contribute to this, and mitochondria don't really. So when someone tells you that cancer must be a metabolic disease and you can fix it by taking sugar out of your diet, that's because the cytoplasmic transfer experiments show it's true. Just remember, they're using 50-plus-year-old data that hasn't been updated. Also, when a doctor tells you that standard of care for your cancer is chemotherapy and that genomic testing isn't worthwhile, they aren't taking into account that you can tell from the copy number variation how fast your cancer is likely to adapt to that treatment, which is critically important for you to know. Understanding the biology of this extrachromosomal DNA changes not just how you think about the drivers of cancer, or I think about the drivers of cancer, but how you should plan your treatment for best efficacy, how you should plan your treatment to minimize drug resistance, to get the best response you possibly can. It lets you know whether to expect your cancer to develop drug resistance and how hard you need to push to contain that. As always, everyone out there is trying to balance between treating their cancer hard enough to get a good response and soft enough to minimize side effects. But if you don't know about extrachromosomal DNA and you can't know how hard you have to treat the cancer to get a good response, you can't make that calculation. You may go way too soft and regret it later. So please go get a genomics panel that includes copy number variation so you can make informed decisions for your health. Please. Thank you, and stay curious. I made a mess up here. I'm going to deal with it later.
Beyond these videos, if you need more personalized guidance or a deeper dive into specific treatments to have your treatment be as effective as possible, I offer one-on-one sessions and medical advocacy. You can find information on our website, which is linked down below. Again, if you found this video informative, please give it a thumbs up, click the notification bell, and subscribe to our channel for more science-based cancer insights.